This is my presentation on gene editing and de-extinction. Gene editing, or better known as a whole as genetic engineering, is considered to be a new and possible way for extinct and or endangered animals to regrow and repopulate their species. And with many species of animals going extinct, some that we know of and some that we don't even know of, and no possible natural way for repopulations to regrow once going extinct, gene editing can make this impossible task seem very possible. Gene editing is when scientists take out a portion of a gene and with their ATG and C nucleotides, which they're made up of, and rearrange and edit the order and sequence these ATG and C nucleotides show up on the gene, which then end up creating a new trait or editing a trait that was already existing. Gene editing and de-extinction are considered biology because by definition, the study of biology is sorry, the study of biology is the study of living things, and gene editing is editing genes, which are the basis or coding for how these living things act, look, behave, and any other trait that they might have. When talking about this, when talking about this topic, I want you to remember three important things, and one of those is being what Colossal Bioscience, the company who was successful in de-extinction, what they did and the science behind it. Colossal Bioscience were effectively able to look at direwolf's DNA sequence when they extracted DNA from a single direwolf's tooth, as well as a direwolf's skull, mainly in the petrous bone, where a large amount of DNA is found. Oh, sorry. Once they once they got this DNA, it was able to look at the sequence of the direwolf's DNA. They then took a somatic cell or non-reproductive cell from a gray wolf, which has the closest genetic sequence to the direwolf. They then took a somatic cell and took the nucleus out of that cell and took the gray wolf's somatic nucleus cell and edited the DNA and genes inside of that. Once once they edited the genes in the series of nucleotides within the nucleus of the gray wolf's somatic cell, they then took another egg cell which had that nucleus removed and then took the edited nucleus and added it into that egg cell, which you can see the process done here in the presentation. Once that new egg cell with the edited nucleus was then created, they then put that in a surrogate dog, which were specially selected for their size and health to be able to carry these direwolf puppies to term. BioLife's Colossal Bioscience has been successful three times on the many attempts they have, giving us three direwolves so far, two brothers being Remus and Romulus, and their sister being Khaleesi. Remus and Romulus are the same age, and Khaleesi is about 3 months younger. The next point I want you to understand is why colossal science used direwolf specifically for de-extinction. According to Ben Lamb, the Colossal Labs Biosciences co-founder and CEO, he said that on all paths to our de-extinction projects, we try to pair the animal we're de-extincting with that of its closest relative or cousin species that is threatened. And they found out that the closest living relative to a direwolf and with a similar genome is a gray wolf. They then took the gray wolf's DNA and meticulously edited it to make to make the gray to have a gray wolf have more dire wolf like traits. They only changed certain traits, however, because according to Chief Science Officer Beth Shapiro, they can't make all the changes because technologically right now it's not feasible, and there'd be many millions of changes that they'll have to make in order to do it all. Instead, they took the traits that they found to distinguish the direwolf the most, like a stockier body and head and a thicker kind of fur coat that you can kind of see in this picture here. And they edited those specific traits in onto the gray wolf, making the direwolf. The main reason as well direwolves was chosen for this de-extinction is because the goal of this was to actually bring attention to a endangered species of wolf that is actually on the brink of extinction called the American red wolf and is considered to be the most endangered wolf on the entire planet. As you can see here on the presentation, that's what a red wolf, American red wolf looks like on the right. I think they're cute. The last point I want to talk to you about is what this means for the future. Even though de-extinction is possible, we're not going to be going around trying to de-extinct a bunch of like ancient birds or dinosaurs or any other older animal. The main goal for de-extinction, however, is to not really help the species that have already become extinct, but to focus on preventing more species from becoming extinct. The focus is for more species conservation and preservation than revival, like with the American red wolf. This is this is a significant step towards this because according to the World Animal Foundation, they estimate over 50% of all species on Earth could become extinct as early as 2050. That is very soon, and hopefully this gene editing can push that date back even further. Another example of this, besides the dire wolves made from the same company as Colossal Biosciences, what using the same method is when they took woolly mammoth DNA and put it in your common mouse. As you can see on the right, the mouse has sort of shorter gray brown coat, and once the mammoth DNA was put into the mice's genome, they developed a more of a thick, wavy, woolly, long hair than their kind of short, straighter hair. The scientists at Colossal were also able to replicate the mammoths accelerate metabolism and also put it in the mouse as well. Colossal Biosciences hopefully hopes they will see a reappearance of the woolly mammoth as early as 2028. In conclusion, with the amazing efforts of everyone at Colossal Labs and Biosciences and the research into gene editing, we have been successful in the de-extinction of certain species, and hopefully this new technology will not only maybe de-extinct species that have already gone extinct, but hopefully this can help conserve and preserve species that are already considered to be endangered or on the brink of extinction. When thinking about all the possibilities of gene editing, what species would you like to see revived if possible? And these are my list of sources. Thank you.